test. Okay. Hi, everyone. My name is Megan. This is Seungbin. We're from the Daughter-Lot Lab at Purdue University. And I just want to go over the projects that we're doing in our lab and how it fits with A3D3. So we have Maria, our PI. There's me and Seungbin in the lab. And then we also have Lizzie and Sam. And I'll kind of go over everyone's projects. So a little bit of an overview of our lab. We are a systems neuroscience lab, and we have two main areas of study. So we're trying to understand how sensory information is processed in the visual and somatosensory systems. And then we also want to apply these insights toward the development of artificial sensation. And so to do this, we use a lot of different techniques. We're doing animal behavior, in vivo electrophysiology, in vivo 2-photon imaging. I go down. Is that better? I'm blowing everyone's eardrums out. Is this better? Okay. Is that better for everyone? Okay. So yeah, we're doing animal behavior, in vivo electrophysiology, 2-photon imaging, and electrical stimulation. And I'll kind of go over what all of those are, along with Seungbin. So kind of a background of the goals of our lab, the research goals. So we rely on sensory information to guide and plan our movements. So if you take this football player, for example, he relies on different sensory modalities in his environment to execute certain behaviors during a football game. One of those would be audition. So he's getting auditory feedback from the crowd, from his teammates, from his coaches, so he can modify his behaviors and hopefully get their team to score a touchdown. He also relies on proprioception. So proprioception is the sense of our body's movement and position in space. So for example, he'll use proprioception with his arm so he can know where his arm is in relation to his body, so he doesn't accidentally throw the ball at the back of his head. He can throw it to his teammate instead. And then he also relies on vision. So he needs to find a visual target, so one of his fellow players that he can throw the ball to. So again, hopefully they can score a touchdown. So he relies on all of these. However, if he were to get injured or develop a disease, some of those sensory modalities can be lost. This can negatively impact his ability to play football. And so that's where we introduce artificial sensation. So the goal of artificial sensation is to replace lost sensory function. And so that kind of moves me into the two main goals of the lab. So first one is to look at natural sensation. So how is natural sensory information encoded within the brain? And then we're also looking at artificial sensation or artificial proprioception, where we can use electrical stimulation to manipulate neural activity within the brain. So that's a little bit of an overview. I'm going to kind of briefly go into all the projects that we're doing. So the first one is with Lizzie. She's doing some really cool work. She just finished her first year, finishing up her first year as a PhD student. So she's looking at traumatic brain injury and its effects on cortical physiology. So with traumatic brain injury, it can range in severity. It can be mild, moderate, or severe. But about 75% of cases are mild. So a lot of them are the mild, and they can sometimes go undetected. In patients with traumatic brain injury, about 90% of them report deficits in the visual system. So they can have blurred vision, a reduction or complete loss of vision, or difficulties with eye movements, for example. And so what Lizzie's really trying to do is take this information that we know about the visual system after TBI, traumatic brain injury, and see if we can use that to estimate what's happening in the cortex in the brain, and see if there's any correlation between the two. And so what she's been doing is setting up this behavioral task with our mice. We use mice as our animal model. And so what she does is head fix the mice, and then she puts a computer screen in front of them. And on the computer screen, she has all these little dots. And she can manipulate the movement of these dots to represent a certain coherence. So coherence, so for example, 100% coherence would mean all the dots are moving in the same direction, whereas like a 40, 30, 20% coherence, they're all moving in different directions. So she's trying to train these mice to report the global motion or the global coherence of these dots. So let's say she gives a visual stimulus of 100% coherence in the right direction of the dots. What she's trying to train the mice to do is move this wheel. You can see they're holding onto a little wheel. She wants the mouse to move the wheel in the opposite direction that the dots are moving. So they have to make this this conscious perceptual decision so we can test how the visual system is working in these mice. And so this is kind of like the natural sensation, what's naturally happening from here. And so she's done a little bit of data analysis with this. one of the biggest things we have to do with behavioral neuroscience is prove that our subject is actually learning the task that we're having them perform, and they're not just doing it by chance. So what she's done here is over 15 days of training. You can see there's only 2 mice, but you can see they generally have an upward trend in their performance, meaning that as the training goes on, they're learning how to do the task, which is what we want to see, obviously. And then she also found that the performance can increase when the coherence of the dots is higher. So, which would make sense. If all the dots are moving in the same direction, it's a lot easier to tell what direction they're moving in rather than if the coherence was smaller. And so she does see a drop off as the coherence decreases there with her mice. So after she's going to refine all of these little things in the behavioral task, she wants to introduce the traumatic brain injury. So she wants to do a mild traumatic brain injury into the visual cortex and look at how the visual system is impaired. And she'll do that by performing electrophysiology. So where you can record neural activity in the brain. And so she wants to record neural activity with electrophysiology after TBI while the mice are doing that same task. She can kind of see behaviorally what's happening and then in the brain what's happening. So it's really cool work that she's doing. I'm really excited to see where she goes. So then we have Sam. Sam's kind of going into the artificial sensation side of our lab. And so if you remember our football player example, so we use multisensory integration such as audition, proprioception, and vision to perform a behavior. But when this is lost, it can negatively impact your performance, your ability to do different behaviors in life. And so that's where we introduce artificial sensation, where you electrically stimulate the brain to hopefully replace that lost sensation. And an example of this is with cochlear implants. So people who are hard of hearing, they can have a cochlear implant to electrically stimulate their brain and hopefully replace that loss of auditory stimulus. So there is some research on this, but there's still really a need to understand how the brain forms these multisensory connections so we can produce a meaningful integration of both artificial sensation and natural sensation. And this will help the development of these sensory prostheses, such as the cochlear implant. And so Sam's doing this really cool thing. Hope this plays. So he's been working really hard on developing this task. So what he's doing is placing mice in a behavioral cage, and he's giving them either a visual stimulus or an auditory stimulus. So here you can see the visual stimulus is an LED on the ground. And so he's training these mice to locate the visual stimulus and go to it. And once they do that, they go to a reward port where they get a juice reward. They get strawberry Nesquik. So he's seeing how they can learn to visually navigate in this field, and then he's also giving them an auditory stimulus. It probably isn't playing the auditory or the sound, but he's giving them an auditory stimulus, and he's seeing how well they learn to perform the task that way as well. So he's kind of comparing visual and auditory stimuli and seeing how these natural sensations, how they help the mouse learn the task. So his data analysis is still ongoing, but he's getting some really cool results. We're really excited to see where that goes. And so once he kind of finalizes that behavioral task, he wants to introduce the stimulation. So he's going to do intracortical microstimulation, where you implant an array into the brain. He's going to look at the somatosensory cortex, which is involved in, sensation. And so he's going to stimulate the brain and see how he can hopefully integrate this artificial sensation and the natural sensation, so the auditory and the visual stimuli. He wants to see if he can integrate the two and see if that produces better outcomes for the mouse to be able to learn this task. And this can hopefully provide insight into how in the humans, how we can better develop sensory prostheses. So then I'll go into my work quickly. So I'm going back into the natural sensation, but I'm also incorporating a motor component into it. So I'm really interested in how proprioception guides multi-limb coordination. So if you remember, proprioception is our sense of our body's position in space. And so we know that humans and animals rely on proprioception and multi-limb coordination to move throughout their environment. So for example, if you're walking on uneven terrain, like the crazy hills here, we have to coordinate our limbs to maintain our balance and continue and overcome any obstacles that might be in our way. We know that the motor cortex and the somatosensory cortex in the brain are both involved in this independently. There's been a lot of research of the independent contributions to multi-limb coordination, but we don't really know a great deal about how they work together to achieve this type of skilled limb movement. You don't really know, like what kind of information are they sending to each other, and how they're working together to do this. So that's kind of what I'm trying to get at. So the experiment that I developed, I'm looking at the afferent pathway. So that's when you take the sensation from the environment and it goes up to the brain, versus the efferent pathway, which is when it's already in the brain and you're sending the information back down to the limb to make a movement. So I'm interested in the afferent pathway and looking at proprioception information. So what I do is I anesthetize the mice and then I provide a proprioceptive limb movement. So I just kind of naturally brush their limbs, and I want to see how that limb movement is represented in the brain. And so I do this with 2-photon imaging, and Seungbin will give a little bit of a description of that in her slide, but I can basically look at fluorescent activity in the brain, which tells us a little bit about neural activity. And so I'm seeing how neurons are responding to movement of the different limbs. And so I've split my analyses into the independent limbs. And so I recorded on the right side of the brain. So anytime I say ipsilateral limbs, that's the same side of the body. So that would be the right limbs, whereas the contralateral limbs are the opposite side of the body, so the left limbs. And so what I've done here is I made a little map of the brain and the areas that I'm interested in. So the motor cortex, somatosensory hind limb, and somatosensory forelimb. And then I showed the imaging area of one of my mice, and all these little black dots are the neurons that I recorded throughout the entire session. So I processed the data, filtered out any neurons that weren't really responding that well. And then I get this map over here. And in this map, it's showing of the neurons in that area, which limb were they responding to the most when that limb was moved. So you can see these light blue neurons, these are responding the most to movement of the contralateral front limb. These red neurons are responding to ipsilateral hind limb. And what's interesting here, so traditionally in neuroscience, it's thought that, and have been proven, that movement on one side of the body is represented in the opposite side of the brain. However, with these results, there are some ipsilateral, so same side of the body, representations of the neurons. So this is kind of further suggesting that the brain can be involved in multi-limb coordination, since there's representations of both sides of the body in here. So I want to look into this more. I have some plans to refine the protocol, the simulation protocol, and go from there. And I'm really excited to see what that gives us. And so from here, I'm being very ambitious, and I want to develop a transsynaptic tracing technique. So I'm basically doing viral tracing in the brain to track neurons in S1 and M1, the somatosensory and the motor cortex. And I want to see what kind of kinematic limb information is being communicated between these two areas. So for example, I want to see if they're communicating, like the angle of the limb, or maybe how fast the limb is moving, different things like that. So I'm really interested to see what I can do there. And then I'm also going to be doing optogenetic inhibition to look at how each of these areas is contributing to locomotion in a novel environment. So optogenetic inhibition, that's when you can temporarily inhibit a certain part of the brain. So I'm going to be inhibiting both S1 and M1 while a mouse is walking on this wheel, and it's a novel thing I'm trying out. It's basically like a ladder, but in wheel form. And when you put the mice on this, it requires them to coordinate their limbs. Remember, we were talking about multi-limb coordination. So it requires them to coordinate their limbs to walk on the wheel. And so I want to see, upon inhibition of S1 or M1, how does that affect their behavior? How does that affect their ability to walk on this wheel? And so I can do some cool behavioral analyses with that. And then I'll also do electrophysiology to see how the brain is responding when that area is inhibited during locomotion. And with that, I'll hand it off to Seungbin.
Okay, it's working. So from here, I will introduce one of our projects about neural decoding using neural network. Neural decoding is decoding neural signal to predict any behavior variables. For example, in our case, we are trying to decode neural signal recorded by 2-photon calcium imaging to predict limb positions of a freely running mouse in video frames. And neural decoding is important to develop applications such as brain-machine interface for patients suffering from neurological injury and diseases. And it's also important to understand the function of the brain. And behavior emerges from interactions of large number of neurons. So it is important to record a large number of populations of neurons at the same time. And two-photon calcium imaging is a promising technique for that. Compared to electrophysiology, like while electrophysiology can record hundreds of neurons, two-photon calcium imaging can record hundreds to thousands of neurons. So Megan briefly introduced this before, but when neuron fire action potentials, there is an increase in calcium concentration in the brain, and two-photon calcium imaging measures neural activity indirectly by measuring that calcium concentration changes in the brain. So not only the relation between neural signal and behavior is complicated and nonlinear, but also getting real neural signal from these two-photon calcium images is one of the big research problems in neuroscience area because it represents real action potentials indirectly. So considering all these complexity and nonlinearity in the problem, we are trying an approach using neural network. So here we are trying to decode limb trajectories from two-photon calcium imaging using a recurrent encoder-decoder network. We are using a recurrent encoder-decoder network, which is sequence-to-sequence model, because one of the main problems in this case is that the frequency of the two-photon calcium imaging is much lower than the frequency of the behavior, which is 7.8 hertz and 30 hertz. So we have separate encoder and decoder with LSTM layers, so we can set the length of the input sequence and the length of the output sequence differently. And in this case, we are expanding the shorter sequence to longer sequence. And input to the neural network is these fluorescence traces extracted from two-photon calcium images. These fluorescence traces represent like fluorescence changes over time for each neuron. It is normal to use these extracted fluorescence traces for using two-photon calcium images, but in the future, we are hoping to use these raw calcium images because there is a loss of information if you use these extracted fluorescence, because for example, the spatial information, like spatial relation between each neuron is disappear in these fluorescence traces. And the size of these images are huge. So we are expecting we need to use some high throughput AI method for that, but it's like future plan. For now, we are trying to use these fluorescence traces extracted in this preliminary research. And the output to the model is these limb coordinates from behavior video. We are using package called DeepLabCut, which is also deep learning based pose estimation package. You can it is pre-trained, so you can just have a few manual labeling, and you can just easily extract predict these limb coordinates in video frames. And in these preliminary results, we confirmed that all limbs, including contralateral or ipsilateral and front and hind limb, could be decoded simultaneously from one model or separately from multiple models, only from two-photon calcium images recorded in single cortical hemisphere. These blue ones are ground truth and red ones are prediction. And this is example video showing predictions. And so we brought these data sets for hackathon and including Megan's experiment data set. So if there is a chance, I will explain this data set in more details. And yeah, that's all. Thank you.
Okay, so thanks to both of you. If you have questions, comments, any discussion, please raise your hand. We can bring the microphone to you.
How often do you need to think about the difference between a mouse's sensory abilities and a human's?
Thank you. Good question. Yeah, it's a really big thing trying to translate animal models into human models. And so it all kind of starts at the animal. And it's a really difficult pipeline, long pipeline to get from animal models into clinical settings. But that's how, we try to plan our experiments to capture human sensation as much as possible. So we really try to consider all aspects of that. It's difficult, but we take every precaution that we can. Yeah.
More questions?
So going back to the TBI discussion in the beginning, you mentioned that you like inject these artificial stimuli to replace kind of the natural ones that are lost. Do you ever, or does your research kind of cover how those also recover, and if that artificial stimuli like helps the natural kind of abilities recover, or is there a connection there? Or?
So I don't think our research really kind of looks at the recovery process of it. We haven't actually like used the artificial sensation experiments yet. So that's still kind of in the process. That is really interesting. There is research on that. I think we're hopefully gonna delve into that a little bit. So to be determined.
Thanks.
Any more comments? Okay, so let's thank Seungbin and Megan again.
So screen, can we do so we'll also want to look at my notes. So we don't have presenter wheel?
I mean, do you have a video? Is there a reason why you can't use a PDF?
Yeah, we have animations.
Yeah, so I don't know how.
Okay, then.
So you can. I can also display your notes on the side here.
Yeah, but it's by slide. But we'll try to see.
Yeah, just here. You might be able to see it.
Yeah. Do you mind? Yeah. Open up the slides from this computer. Sure. We sent it to Melissa. Well, we haven't uploaded yet. We haven't uploaded. Yeah, I want to send it to Melissa. Okay, so let's present from our slides and see how it goes. But there's yeah. Yeah, that's PDF. The one that do PowerPoint. The one this one here. You need access USB to a computer. Oh, you want to do the fancy switching. Yeah.
Would you like this one? Yeah.
Please start.
I will just hold the mic.
Okay, and also.
Yeah. Or there's the small one. The flip one. The skip thing. Yeah, I'm sorry.
Okay, so next we'll have a talk from Amy's group and UW. So Lauren and Shija, they are going to share their presentation. Okay, let's get started when you're ready.
All right. Hello, my name is Lauren Peterson. I am an A3D3 postbac. I am presenting with Sajia Lee, who is a PhD candidate, and we will be presenting our research that we and highlighting some of our research that we have been doing in the Orsborn lab. All right, to motivate some of our research, we're going to start broadly. Technology allows us to expand our capabilities. For example, we are using a computer to present this research to you. Many of you probably used a car to arrive here either yesterday or today. Interfaces allow us to use this technology by allowing us to control it. We control technology through interfaces. For example, we're using a clicker device or a mouse to control this keyboard. And we're using this. Oh, okay. And you use a wheel to control a car. Now, when we think of interfaces, we usually think of interfaces that we control physically with our hands or our feet. However, there are interfaces that we can control with the brain. For example, if we use implantable electrodes, we can use neural activity, record the neural activity, and use that neural activity to control an interface through a computer. One example that we will show here today is a brain-machine interface that can actually restore movement to people with paralysis. This woman has boxes above her head. These are the electrodes that are implanted into the brain and that record the electrical activity. Those wires connect that electrical activity. The wires connect the electrodes to the computer, which can then process and use that neural activity to control this device. And then we use the electrical activity to actually allow this woman to feed herself a chocolate bar. So with this technology of neural interfaces, there are a huge range of applications and implications. We can revolutionize healthcare with these cochlear implants, restoring movement, a retinal implant, or deep brain implants to treat neuropsychiatric disorders. I'm going to talk a little bit about the cochlear implant. We implant electrodes into the cochlea so that it can record the activity, that it can record sounds. And then we send that to a processor to interpret the sounds and restore hearing for people who have some hearing, who have loss of hearing. This also allows us to revolutionize human-computer interfaces, and there are ongoing research projects by a lot of big technology companies to this effect. But in order to actually work on neural interfaces, this requires interdisciplinary engineering. We need hardware and materials to measure and manipulate the nervous system. We need algorithms to interpret neural activity. We need devices that can dynamically interact with users. And we need to balance the security and ethics so that we are handling all of this in a responsible and ethical way. We also need high throughput and real-time AI, which is where a lot of A3D3 comes in. We've had a rapid increase in number and type of measurements over time. The y-axis is a log scale, so we've been increasing exponentially over time. We need a data-driven discovery of relevant features and structure in the data so that we can minimize computational load and increase latency, or decrease latency, increase performance. We also need algorithms that interact with the brain and control devices in real time. What we know for sure is that any delay in the algorithm and decoding algorithm actually kills your accuracy. So you cannot control this algorithm or device with any accuracy if there is a delay. So we need low latency algorithms too. How do we make machine learning that interacts with the brain? We need, much like you learn to ride a bike, your brain learns to control a neural interface. Machines can also learn and adapt to aid this process, but assuring both learners work together requires understanding the intersection of neuroscience and machine learning. This is work being done by Manishka Maduri, a student in the Orsborn lab.
Okay, so first of all, I'll talk about what are the different type of neural signals that we can record from the brain. So let's look at here. From the surface of the brain, we can record what we call echoic signal, essentially the continuous measurements. So this is not working. Sorry about that. From the surface of the brain, we can also put electrodes into the brains, for example, the neural pixels. From which we can record local field potentials, which are also continuous measurements. And from the same intracortical electrodes, we can also record action potentials of neurons, which are discrete measurements like zeros and ones. And so each type of recording actually gives rise to many types of neural features. A really good example is the ECOG signal, which are broadband signals, and you can do FFTs and get all these features in different frequency bands. So with different types of recordings in our lab, we have five monkeys in our lab, and they're engaged in different tasks. And this is a spectrum of tasks and data structures that we have in our lab. So on the left-hand side of the spectrum, we have the more constrained lab environment, where the monkey is engaging in a mentor-centered reaching task. And because they're constrained in a lab, we can record many different types of behavior data, for example, eye tracking and motion tracking information. And in the meantime, we can also record brain signals from these monkeys. And this is what we call BMI centered reaching task. Excuse me. On the right-hand side of the spectrum, we have the last constraint in environment, where we can record unstructured data, where the monkeys are performing these tasks inside a cage, where we can get wireless recordings. So these are the particular questions we're interested in. Well, we can record my different types of features. For example, the first couple of questions are what are relevant features in electrodes, and what are the underlying dynamics, as well as in a real-time BSI experiments, we'll have to select what are the electrodes to stream as the user is also trying to learn the device. So with these objectives, the conventional machine learning algorithm doesn't account for these non-stationary symbol distributions as the learner is trying to learn the device. So we developed this convex algorithm to account for the sparseness, smoothness, as well as relevance when selecting features. So the first objective here is to minimize the number of features as the subject are the monkey trying to learn a task. And the second, the second term here is to maximize the feature selection smoothness. So this is a key innovation in our work, trying to make sure that while the user is trying to change, we're also selecting consistent features. And last one is a term that's very similar to loss regression, where we measure the feature relevance by measuring the weight to noise ratio. And this work has been done built on previous convex optimization algorithm that allows to solve this objective in real time. And this is our work in preparation. All right, so we did some simulations with these objectives, and the ground truth here basically shows, well, we're trying to flip the features at midtime of the simulation, and the ground truths are the feature we want to select in a real-time simulation experiments. And we compare the performance of our convex algorithm to loss regression, as we can see on the convex for the convex algorithm, we can select consistent features across the simulation. And for the loss regression, we can see the spiky features across the feature selection batches. And we compare the performance of those two different types of algorithms. And the blue line here is the convex algorithm, where we can get more rewards as the brain is trying to do the scenario reaching task. So this is all in simulation. So next one is trying to move from our simulation work to actual animal experiments. So this is all real time. We're trying to implement our algorithm in animal experiments, in actual scenario reaching task. And we're also interested in how do we do ECOG feature selection by using data-driven selection methods. And Lauren is going to talk about another type of work in modeling.
All right. So Sajia has been working on how do we use data to determine which features are the most relevant? What if we were also able to build a model of neural signal dynamics and effectively model the brain and understand and verify these results that we have? This is based on work from a preprint by another former student in the lab. Multi-block RNN autoencoders enable broadband ECOG signal reconstruction. So modeling neural dynamics, we have recurrent autoencoders. These current models have been useful for neuroscience research. However, they have also mainly been used for spike data, where you have an input of ones and zeros going in and then an output. You compare that output, predicted output, or the reconstructed output to the given input. But we also have ECOG signals, which are broadband. What happens when we use a broadband signal instead? If we make some minor adjustments to handle the broadband, we get this RNN autoencoder model, or an RAE. We also know from preliminary experiments that this RAE is able to reconstruct signals with some accuracy, but the accuracy actually decreases as we ask it to reconstruct higher frequency signals. Those higher frequency signals are known to correlate with a lot of movement activity, and so we want to keep those signals. We want to keep that accuracy and that information. If we increase the model size, we increase performance, but increasing the model size is actually not scalable. So what now? This previous student's innovation was to have a multi-block RNN autoencoder model, where we separate each frequency band and give it its own autoencoder model. Therefore, every frequency band is handled independently of the others. And then we combine them at the end to reconstruct the entire broadband signal. Performance improves with model size again. The advantage that this model has is that it is very scalable. So you can have many, many frequency bands and many, many blocks, and that maintains accuracy in a way that just having a single frequency band autoencoder model does not. So for some real-time applications, FPGA implementation is in the works. We are collaborating with the Shlizerman and Hauk labs to improve performance and latency, and they will be showing you some of their research later today. We know that the MRAE is scalable, but other emerging technologies, such as transformers, might improve reconstruction and prediction and help us determine which features are the most important so we can evaluate and compare to some other results in the lab. We would like to thank everyone in the lab and NSF and A3D3 for funding this research. Thank you.
Thanks. We have lots of time to discuss and for questions. So any questions?
So I'm not sure if I understood it right, but I think the goal of the reconstruction of the neural signal is to get better representation of real neural signal. And what is the ground truth of your reconstruction model, and what is the performance?
Yeah, so the data that we're using for this experiment is the data collected from the monkey in a completely unconstrained environment. And we have hours and hours and hours of that data. And so what we do is we feed that data into this algorithm. And then when it reconstructs the data, we compare it to the original data.
Same one?
Yeah, we were trying to minimize the mean squared error between the reconstructed data and the actual data.
You go back to the slide on MRAE.
Yeah.
The FC at the end, how much inputs is that getting and how complicated is that FC?
So I don't know that off the top of my head. It is getting the input where we feed all of the input from all of the frequency bands.
From all the MRAs?
Yeah.
Okay, so a huge input, moderately small output. Do you know, is it a neural net? Is it a summation?
I think it's a neural net.
This the FC, is it like fully connected something?
Yeah, fully
Okay.
connected layer.
Okay. More questions, comments?
So you said that the performance improves with the model size. Around what model size would you consider to be good enough? I guess how many parameters?
So a lot of this work was done with 1536 features for the 3-block model. So that would be about 500 for each frequency band. And the performance did improve between having a single block that was 500 and then having 3 blocks that were each 500. I don't know if that was considered good enough. I think we would need to have more blocks.
Okay, thank you.
Thanks. I have one question. If you go back to previous slide. So you say here that if you increase the model size, it improves, but then it's not scalable. So what exactly that means? Like,
Yeah, so this is an example where you would have, I like to think of it as having everything in series instead of everything in parallel. So the bigger the bigger the encoder decoder, the bigger the neuron size, the bigger the model size, and it just takes more time.
okay,
to
and
complete.
do you have any like time latency requirement that you want to achieve?
Not right now.
All right. Now. These are all like post processing. So you collect the data and then you analyze them later on.
Yeah,
Right.
we've already collected all this data.
Okay. More questions?
So I just have like 2 questions that you're using, like of an MS buffered in any way based on the data that you're collecting.
Buffered how?
Pre-processed.
All of this data has been pre-processed to remove as much noise as possible, but I don't know if the MSC has been adjusted for this data.
Second is when you try to use neuropixels, how accurate can you get them in like the same region of monkey A?
So we haven't done a whole lot of neural pixels yet. I can't answer that question yet because we don't have a lot of neural pixels data.
Okay.
All right. It seems like we don't have any more questions or comments. Let's thank Lauren and Shicha again.
Hello. This might be easy.
Hello, everyone. Today, me, Yang, and Chuang is going to present our research happening in the neural AI group. As you see here, we are the data-driven dynamic system research group led by Professor Shlizerman. We are supported by the electrical and the computer engineering department, also the applied math department. So, as you see here, in our lab, we focus on two parts. One is neurobiological networks, and the other is artificial neural networks. We use the methods to analyze the spatial temporal data like machine learning and dynamic system methods. And our lab mates are working on the AI algorithms and neurobiological networks on the actual organisms. Here is an overview of our trainees. I will just leave it. So a brief introduction of our lab's project. Specifically for the neural part, we proposed the spatial temporal data transformer, and also in collaboration with Orsborn's lab, we proposed the multi-block autoencoder. And for the AI part, we proposed the Lipton-guided embedding for hyperparameter search. So in this case, you can decide the predict the model performance based on the Lipton-guided embedding. And the other work is for the scalar-based action recognition with active learning, which can be applied for the neural, like animal behavior as well. Next, Chuang will give the introduction of the neurons.
Yeah, thanks, Jingyuan. So I'm gonna give a brief overview about the recent research in our lab concerning the developments of algorithms to process neural time signals, especially for high throughput and low latency applications. So we know the brain encodes the sensory stimuli and motor behaviors in its spiking activity, and this activity spans across time and space. For example, we can have a okay. So this is an example of the spiking activity, where we have the x-axis as time progression, and the y-axis is the number of neurons in the population. And to understand this dynamics that is going on here can help us relate the neural activity with the behaviors of animals, including humans. One example of which is that we can record the neural activity and decode the motor intent of the humans and translate it into actual handwritings. But the problems of the neural decoding is that the activity of the neurons are intrinsically noisy. So here we show a snapshot of a smooth version of this spike trends over here. And as you can see, it's very noisy. Even when the neurons are in the same behavioral conditions. So the problem is how can we decode the behaviors from these noisy ones? It's in fact very difficult. So we need algorithms that could denoise these neural signals and figure out the underlying dynamics, based on which we can do the neural decodings much easier. So I'm just talking about what I mean by neural signals. So based on the applications and based on the recording modalities, we can have several kinds of neural signals. If we have the electrodes sticking into the brain, we can record the actual spiking activity of the neural populations. We can smooth it out and use the firing rates over time as a neural signals. And if we have the electrodes placed on the surface of our brain, then we can have the echoic signal. And if the electrode is outside of the skull, we can get the EEG signal. So all of these signals can and need different algorithms that are tailored specifically to accommodate its unique characteristic. Also, people in the community also collect a lot of data and organize some benchmark so that we can test our models and compare between different approaches. So here we have an example of such benchmark. This is called neural latency benchmark, and it is collected across multiple animals that are doing different tasks, and also the recordings are from different regions with a wide variety of neurodynamics. So to process all these large-scale neural data, people have turned into artificial neural networks to modeling for modeling the underlying neurodynamics. One famous model is called the alphas model. This is a recurrent neural networks in the framework of a variational autoencoder. This performs is for a long time has been a state-of-the-art model to infer the underlying dynamics. But the problem with this INN-based model is that it has to process the neural time series in sequence. So this will obviously cause some unwanted latency when we want to deploy these models into real-time applications. So people have proposed transformer to do the same task. So the advantage of transformer is that the transformer can process the data in parallel, and it has been shown that this could speed up the inference up to like 6 times compared to alphas. But on both of these models, NET and alphas, they only model the temporal dynamics, and which I will talk about the disadvantage of this one. So we know that the brain, the neurons in the brain, do not fire independently. It has been shown that the different neurons have different kind of tuning curves, and the activity correlates with each other. So collectively, all of these neurons represent the motor behaviors, and it's necessary to take this into account when we build such a model. So in our group, we have proposed a spatial temporal transformer, which explicitly model this temporal progression and the spatial co-activation of the neurons. Our work has been published in Neurops last year. So we show that by using this spatial temporal transformer, it has outperformed on other state-of-the-art methods in recovering the underlying dynamics of the neurons. The state-of-the-art includes the alphas and NDT that we just told before. Secondly, it has by modeling both the spatial and temporal dynamics, it has recovered the stereotypical responses of the populations by just a single trial. And lastly, we can, which is the main thing, we can decode the motor behavior from the output of this model. And we can show that the decoded motor behavior very accurately resemble the truth behavior. The last thing we can, from modeling the spatial dynamics, we can get some insights into how the neurons in the population collectively encode the behavior. We see that the SDNDT model has revealed a subset of neurons, which are the bright column here, that are mainly responsible for driving the dynamics of the home populations. And if we drop out these important neurons, then we can see a significant reduction in model performance compared to if we keep everyone's in the same population. So next, Jingyuan will be back to talk about our recent efforts on modeling ACOG data and the use of graph neural networks.
Thanks for introduction. So in the following projects, we focus on the ECOG data sets, which have been previously used for the speech and movement decoding of paralyzed patient. But in our lab, we specifically focus on the ECOG data collected from the monkey motor cortex in collaboration with Orsborn's lab. So here is a visualization of the ECOG spatial arrangement. So each point here is the electrode, and we can visualize the ECOG signals in the frequency domain and the temporal domain. With this data set, we're focusing on the two parts. The first one is if we can reconstruct the signal and understand the signal in that way, and the other part is the forecasting of the neural signal. So for the reconstruction, as previous presentation mentioned here, so previous students, Makin proposed for the MRAE. So this is mainly focusing on the reconstruction of the neural signal, which original broadband signal. So specifically for one frequency band, we can see here, the model is composed of the encoder and decoder and a fully connected layer. So this encoder part is a JRU, which is our kind of model, and also for the decoder part, we show the broad like this. With this decomposition of the signal, we can do a better reconstruction of the original ECOG signal. So the next part, I will introduce the ECOG signal forecasting. So for the forecasting, the reason we want to do the forecasting is like the brain signal, it follows the temporal causality. So with the forecasting, we can like do the similar thing at the brain signal generation. So another thing is like by doing the forecasting, we can reduce the latency for the real-time application of like BCI application. Specifically, as Tron mentioned earlier, so the neural signals show some spatial interactions between the neurons of the ECOG channels. So here we propose to use a graph convolution network to do the neural signal forecasting. Specifically here, we treat each ECOG channel feature as a vertex of the graph, and the edge here describes the connection strength between the channels. And in our proposed method, the edge here, the connection strength is learnable. So like to do the forecasting here, we proposed like to use 3 different blocks, like to do the temporal encoding, spatial interaction, and the temporal readout. For the temporal encoding here, each ECOG channel is modeled separately or independently. The model can be the RNN transformer, any kind of model like you can use to model the temporal signal. And the other part, after the modeling, like extracting the feature of the temporal signal, we use a spatial interaction. Spatial interaction module, which is composed of graph model to allow the interaction between the channels. And after that, we have an additional temporal readout layer, which is also an transformer kind of model. So with this model, we show like our forecasting accuracy, like in terms of the R-square or the correlation of the mean squared error. So our methods outperform the other like transformer or the RNN-based method. Especially if you look at the forecasted signal here, we can see like with the AMAC, our method, like the red curve can catch the big jump here. So which is the ground truth is showing blue dashed line. And other method like LRN or the NET may not be able to catch that big jump. So another thing, as we mentioned, like the edge of the connections is learnable. So we map the ECOG leacock electrode to a 2D array and look at the specific target channel, like how the target channel is connected with other channels. So the darker the color is, means the stronger the connection. So we discovered that we could discover that the target channel is more strongly connected with the nearby channel, which it agrees with previous discoveries. In addition, we also look at the neuron trajectories. The left figure here is a neuron trajectory from the original neural signal, so it's quite messy. The blue dot represents the start point, and the orange is the movement. So we can focus on the blue and the purple curve, like which is start and end of the trial. So it's quite messy, but with the learned model embedding, we can see it automatically aligns the neural signal from start to end. So most of the previous introduction of our research is focusing on the spatial temporal modeling, and most of the temporal modeling is done by the RNN kind of structure. In the next part, Yang will give introduction of the RNN.
Yeah, thank you for the introduction. So you might notice that all those projects they've been using RNN a lot because RNN is very good for the sequential data. But the one problem with RNN is like there are so many options, so many RNN variants. So how do you know which one is the best one for you task? So that's why I want to focus on the RNN stability and the robustness, so that you can, among all the choices, you will know, okay, maybe this one is actually the best one for my project. So yeah, you guys probably like familiar with this like RNN architecture, but I just want to do a very quick recap. So RNN, you have the hidden state H and the input every time step X. So then there's a recurrent matrix, which is the V here. So at each time step, you update the hidden state HT based on the previous hidden state and the current input. And then you have the output of this time step OT. So, but one problem with RNN is the stability problem, which is the exploding and vanishing gradient descent. So because we need to do the backpropagation, which in RNN is like backprop through time. So if you if the gradient like is vanishing, then for the very beginning time step, it's very hard to update. And if it's exploding, then the model just become very unstable. So in one of the previous work, people realized actually we can think about RNN as a dynamical system. And in dynamical system, people use a method called Lipton exponents to capture the expansion or contraction rate of each state. So we can think about like each hidden state as a state in the dynamical system. So then we can use the Lipton exponents to capture that. So like on the right side, we use different hyperparameter configuration for the LSTM, and then we have like different like Lipton spectrum. So Lipton spectrum is like the collection of all the Lipton exponents of the model. And if the Lipton exponents is bigger than 0, means that direction is expanding, otherwise it's contracting. So we have like different like Lipton spectrum for different network. And we can use that to predict the performance of that network based on, so we use an autoencoder. So the input here will be the Lipton spectrum. And we try to reconstruct this Lipton spectrum, use this autoencoder. But differently, we have a predicted network, which is actually just like a 1 or 2 layers of fully connected network to predict the accuracy of that particular model based on the Lipton spectrum. So as you can see on the right, I mean on your right, on my left, so you can see according to the latent space, there is a clustering, like, so all the green dots are the good performance network and red dots are the bad performance network. So just based on this latent space, we can actually see we can like do the clustering just based on the Lipton spectrum. So as I mentioned before, there's so many like variants, and so the problem is how can we choose the right one, and can we use this method to generalize to all the RN variants. And according to this result, we have been using like all the RN like variants, LSTM, GRU, like ASRN, couple RN. And so on the right, you can see all the RN are distributed on like all the space. But we also do the clustering based on the accuracy. As you can see, like just based on the accuracy, they are actually no matter what kind of RN variants they are like most of them are clustered on one side, and so they are the bad performance model are clustered on the other side. So that means this method is able to like generalize to different like RN architectures. And not just that, we want to actually predict the performance of that particular RN like ahead of time. So we also do the prediction, like, so even just with like 10% of the training. So for example, if the model need to train like 10 epochs and this model just train like 1 epoch, and based on the Lyapunov exponents, we are able actually to have a like very good clustering of the good performance model on one side and bad performance model on the other side. So yeah, that just means we are able to select the like a particular RN model ahead of time for you task. So yeah, that ends our presentation. I just want to thank all the organization for supporting and funding those projects. And we have the QA time now.
Thanks. So any questions, comments?
Start with the stupid question. So can I view ECOG as a more precise EEG, or is the data fundamentally different between the two?
Thank you. That's a great question. I may not be very very familiar with the EEG data, but like EEG data is collected out of the cortex, so there could be a lot of noise in that signal, and also for the ECOG data recorded, which is recorded intracortical, so the signal could be more precise. And also you can think about the spatial resolution of the two different signals. The EEG may like record it from the multiple brain areas, but for ECOG in this case, what we are using is the special, which is very small, like this, like the new, like, so the spatial resolution, there will be higher spatial resolution. Thank you.
Thanks for a great presentation. I was curious, how do you identify the most important channels for ECOG or most important neurons when you're doing spike analysis?
Yeah, so the important neurons, we use a transformer, right? And we have two separate attention models for the temporal attention and the spatial attention. So the way we identify these important neurons is that we look at the attention matrix, and we can see that some neurons that are heavily attended by all the other neurons in the populations. And yeah, that's how we know that that particular neurons are like playing some vital role in like driving the dynamics of the whole population.
Cool, thanks.
That's a great question.
This is a question about the Lyapunov minimization scheme. So I'm wondering what part makes this specific to recurrent networks as opposed to something much more general?
So yeah, so we're saying like this part. So yeah, so we using the like the hidden state, like the Jacobian matrix between the hidden state to calculate the Lyapunov exponents. So there's a previous work by Ryan Wolt. He submitted like 2 years ago. So yeah, he was using the Lyapunov exponent to capture the expansion and contraction rate of the like hidden state like over time. So then he calculate the Lyapunov exponents for each state. And then so for example, if you neural network has like 400 hidden state, then there will be 400 Lyapunov exponents. So yeah, this formed the Lyapunov spectrum on the right. So then we use this as an input for the autoencoder for the like the following task. Yeah, does that answer the question?
And just a follow up. And so the expansion and contraction in this context, like you have RNN, like what does expansion mean here? Like just, if you can explain more.
Oh yeah, so if you look at the Jacobian matrix, then so actually we do the QR decomposition, then the Q matrix. So if you see on this side, so at first, for example, this is just like a toy example. So at first, each of the Q1 and Q0 direction, they are like the new unit distance. Then for the next time step, the red one is like shranked, but the blue one is like expanded. So it's like the so it's kind of like the eigenvalue eigenvector. So the eigenvector direction is like expanded and contracted. So
okay,
yeah.
I see. And in your next slide, when you have this autoencoder thing, so here you're just giving all the Lyapunov exponents corresponding to hidden states and try to reconstruct them. What exactly was the task here?
Yeah, so for this task, the input is like the Lyapunov spectrum of each network model. So we are given the Lyapunov spectrum of the model and also the accuracy of that model. So to do the supervised training. So there are two like loss, like the loss in here. So the first one is the reconstruction loss for the autoencoder. The other one is the prediction loss for the predicted theta model here. So then we just like you use a weighted sum of those two losses as the final loss for this task.
Thanks. Any more comments or questions? Still have a few more minutes if you have anything.
One question about the AMAC model. So you showed that the performance is better than the other model. Have you compared, or do you have the number that how complex the comparison of complexity? How much more, I don't know, number of parameters or how much more complex?
Yeah, I see. Thanks for the great question. So the question is like how the number of parameters affect the model performance here. So actually for the AMAC model, the number of parameters is not, we didn't like calculate the number of parameters, but we use a relatively small model compared to other models. But the thing with like the graph network, it takes a lot of memory. So so even though the model architecture is relatively small, but like computation takes more time. And so that's a trade-off between the two. So the graph model is more like provide a prior knowledge of the data. So it can do the better job, but the side effect is like it may take more memory. But the model parameters is not like we don't need a lot of parameters of the model. Does that answer the question?
Okay, thanks. Let's thank the speakers again.